Good afternoon. I am audible now?
Yes, sir. Good afternoon, sir.
Good afternoon. From last class, anyone has any doubt? Because today we'll go for next stage. Anyone has any doubt? During the light reaction, that jet scheme, no doubt.
Sir,
Hmm.
sir, the cycle you told, the Koch cycle, sir, that was my. I'm not clear in that cycle, sir.
That is that water splitting complex. That manganese splitting complex, that is also known as Hill reaction or that all Koch cycle. How water is splitting takes place.
Hmm, okay, sir.
Any other? Hello. Any other? No doubts. So Donesh, Sai Preeti, Samiksha,
Yes, sir.
any doubts? Because yesterday was holiday, na? I think might have read. Arpita, no one has doubt. Okay, then that is very good. Ruplin, Ruplin,
Yes, sir.
you have any doubt?
No, sir.
Have you read or not? No one read. See, while I am teaching, na, some things I intentionally left. That is for you. If you don't ask, na, if any books you read, na, you might have gone through it. That sir did not tell us that things. So by that way, I will understand that who has read or has not read. Okay. Today we'll start that second step of reaction photosynthesis. That is called, what is that? Last class we have read the light reaction. I am audible na clearly?
Yes, sir. Audible, sir.
Okay.
Yes, sir.
Last 2 days we have learned what are the pigments available, what are the role of the pigments, what are the different photosynthetic machinery is available there, and after that we have studied how photon is absorbed, and in with the presence of photon plants split water and release the oxygen to the atmosphere. These things we have learned. So there are two types of that things we call photophosphorylation, because in this process we received ATP or energy. So last slide, I think this one. So what happened? In this process we get molecular oxygen, NADPH and ATP molecules. What actually plant did? Plant absorb photon, split the water. From the water, he released the O2 to the atmosphere and absorb the electron from that water, which transported through photosynthetic electron transport chain. That is different complex system. This one, different complex system from pH2 to pH1 via cytochrome B6 complex, and in this process a proton motive force is generated due to proton gradient across the lumen and stroma. Where is the proton gradient? Proton gradient generated across the lumen and stroma, because in between the stroma and thylakoid lumen, thylakoid membrane is there. So membrane is the barrier between the stroma and lumen. So when both sides of the membrane number of proton is different, it gives a generated proton motive force, which activate the enzyme ATP synthase or proton pump, in which proton will pump out from the lumen to the stroma, and the stroma ATP and NADPH are produced, and this is the end product, that is the which is utilized in the light dependent reaction, independent reaction, or earlier which was known as light dark reaction. This is also known as PCR cycle or photosynthetic carbon reduction cycle. It is also known as PCR cycle. See, PCR you learn biotechnology, that is polymerase chain reaction, but here it is different. It is called PCR cycle or photosynthetic carbon reduction cycle. It is also known as Calvin cycle, because by the Calvin discovered that cycle, and it is also known as CBB cycle, Calvin, Benson and Bassam cycle, because all these three scientists got Nobel Prize for the discovery of light independent or carbon reduction cycle. So all these are same name, so should not be confused with that carbon reduction cycle, carbon fixes, photosynthetic carbon fixation, photosynthetic carbon reduction, CBB cycle, Calvin cycle, light independent cycle, all these are same. So in this chapter or in this dark reaction or light independent reaction, we learn how carbon dioxide fixed and produce the sugar. If there is a lower concentration of CO2 in the atmosphere, what will happen? How these things become controlled by the plants? What are the different factors responsible for this cycle? Apart from that, we learn different types of carbon reduction mechanism. These are C4 and CAM. C4 cycle and CAM cycle. We will study in the subsequent classes. So from the last class, we know this is called Z scheme. Why it is not Z scheme? Because it looks like a English letter Z. So this is the light reaction. We know first what happened by absorbing the photon, pH2 Reaction center chlorophyll activated and transferred to the thiophytin. Thiophytin to quinone A to quinone B, quinone with the plastoquinone. Plastoquinone, they have entered the cytochrome B6F complex. In the cytochrome B6F complex, there is three parts. One is cytochrome F, iron sulfur protein, and cytochrome B. I already told in the last class. And one electron move to the plastocyanine, which is a mobile electron carrier, which pass the electron to the PS1. And now electron pass takes place. When it moves like unidirectional, it is called non-cyclic photophosphorylation. In the non-cyclic photophosphorylation, it is also known as Z scheme. And in the non-cyclic photophosphorylation, NADPH is produced. Then what is cyclic? In the cyclic photophosphorylation, that ferredoxin, after receiving the electron from iron sulfur protein of PS1, it is also a mobile electron carrier. It transfers back the electron to the cytochrome B6 complex. So electron moves within the PS1. So it is known as cyclic photophosphorylation. This much we already know. Now in the Calvin cycle, the ATP produced and NADPH produced in the light reaction used to convert carbon dioxide to sugar. Used to convert carbon dioxide to sugar. Clear? The ATP, why there is a light, it is known as light independent reaction? Because there is no direct involvement of light. But if light reaction not takes place, that plant will unable to do dark reaction. How scientists discover that? Because one dark adapted plant means that plant which does not expose to sunlight. That means light reaction not takes place with that plant. What scientists did, they supplied externally ATP and NADPH to that plant. External supply of ATP and NADPH help for synthesis of sugar. That means that is not directly dependent, and it is also not the dark reaction. It does not mean it is dark. It may also happen in the daytime also, may also happen in the night time only. But it also need the indirect effect of the light. Because after the light reaction is carried out, ATP and NADPH, adenosine triphosphate and nicotinamide adenine dinucleotide phosphate is converted to the sugar. Okay, and this carbon dioxide enter in the cycle, and the leaves they produce sugar. So how it is produced? It produced in the stroma. This cycle is going on in the stroma of the chloroplast. Where it is going on? Stroma of the chloroplast. And in the eastern, what happened? That first CO2 enter in the cycle and it produce a 3 carbon compound. After 3 carbon compound, again one by one 6 carbon added one by one, and other molecule again regenerated. So basically, what happened? This is the brief of that cycle or C3 cycle. We know why it is not C3 cycle. What happened? Cell have already one substrate that is called ribulose 1,5-bisphosphate. That cell already have. How much amount of RUBP is present in the cell? That's the rate of photosynthetic dark reaction or light independent reaction. So what happened? That CO2 and CO2 added with that ribulose 5-phosphate. As we know, this is a 5 carbon compound. 5 plus 1 carbon, it becomes 6 carbon. and after that 6 carbon splitted into 2 3 carbon compound. This 5 carbon ribulose 1,5 bisphosphate sugar added with 1 carbon dioxide, which is added to the RUBP, is 1 carbon, 5 plus 1 6 carbon splitted into 2 3 carbon compound. That is called 3-phosphoglycerate or 3-phosphoglyceric acid. So as CO2 is added, the process is known as carboxylation. This reaction already we have studied in the previous class. Before starting to the module, I have taught now that what is carboxylation. You remember or not? Hello?
Yes, sir.
You remember now?
Yes, sir.
That carboxylation means addition of CO2. So as the first compound is a 3 carbon compound, stable compound, so it is known as C3 cycle. Then if this CO2 is added every time, what will happen? The RUBP pool will finished. If RUBP finished, cell will not able to synthesize that much of CO2. So what this means? Cell again have to regenerate that RUBP during this process. So for that, next step is the reduction. Why reduction means? Dehydrogenase, that is the NADPH. We split it. That NADPH is coming from that light reaction or jet scheme. That NADPH used and it is produced a triose phosphate. Now the CO2 is converted back, transferred into the triose phosphate. This is called reduction. And again, this triose phosphate again synthesize 5 carbon compound ribulose 5 phosphate. So if 6 molecule of RUBP, 6 molecule of RUBP attached with 6 molecule of CO2, it will regenerate 6 molecule of RUBP, and that 6 CO2 will produce some hexose sugar. So this is the basic steps of C3 cycle. There is 3 step: carboxylation, reduction, and regeneration. Okay. So in that, before going to that C3 cycle, you have to remember one key enzyme that is called Rubisco. Full form is ribulose bisphosphate carboxylase and oxygenase. Because this enzyme can have work two functions. One is carboxylase in presence of CO2 and oxygenase in presence of O2. So this is the name of this enzyme, ribulose bisphosphate carboxylase and oxygenase. Why ribulose bisphosphate carboxylase? Because it acts on the substrate ribulose bisphosphate. Means when CO2 is present, RUBP add CO2 and RUBP, and Rubisco add oxygen to RUBP when it is functions as oxygenase. So this is the key enzyme and the largest enzyme and the most abundant enzyme on the earth. So this plant has a large two subunit, or is large subunit and small subunit of 50 different multimeric proteins, and 8 large catalytic subunit and 8 small catalytic subunit together form a whole enzymes. Some bacteria contain only large subunit, but in the higher plants, both large and small subunit are present. One subunit is synthesized from nuclear gene Another subunit is synthesized from chloroplast. So lye subunit within the Rubisco are arranged as a anti-parallel dimer. This is the that you know the protein structure, alpha helix, beta petencil structure. So during conformation, it arranged in such a way the active site will present with some actively amino acids. These are lysine, arginine, threonine, glutamate, asparagine. All these are some charged amino acids which present in the active site. This help presence of this lysine is a basic amino acid which has epsilon amino groups. It can help to react with CO2 or bicarbonate and makes to release the water and proton. So it helps to catabolize or carboxylation reaction. Thus the enzyme helps in the carboxylation reaction. Due to presence of catalytic amino acids or charged amino acids like threonine, asparagine, glutamate and lysine. Okay, and in this process magnesium is an essential nutrient and minerals. And this when that a configuration is there, this is the Rubisco become active or inactive forms. What happened? I told that in Rubisco, usually their active site there is a lysine amino acids which become carboxylated by addition of CO2 and it remain the inactive form. Unless it added with some magnesium ions, it become active. Now it can bind with the Rubisco. Now it can bind with the RUBP, ribulose bisphosphate substrate, and involved in the carboxylation process. First step is the carbamylation. Carbamylation means addition of CO2 to the enzyme, and carboxylation means transfer of CO2 to the substrate. Don't confuse the two terms. One is carbamylation, another is carboxylation. So don't confuse with that. Carbamylation is addition of carbon dioxide to the enzyme, and carboxylation is addition of carbon dioxide to the substrate. Then we know why it is called C3, because the first stable product is the 3-phosphoglyceric acid, which is formed due to addition of CO2 to the substrate ribulose bisphosphate. And this is the steps by which the carboxylation takes place. In this way, we get a intermediate 6-carbon compound. See, RUBP a 5-carbon compound. When addition of carbon dioxide, it forms a intermediate 6-carbon compound. That is named 2-carboxy, 3-keto-arabinotol 1,5-bisphosphate. 2-carboxy 3-keto-arabinotol 1,5-bisphosphate. This is a unstable intermediate. It immediately after this addition, it immediately become hydrolysis and produce 2,3-phosphoglyceric acid. It's split in the middle and it will produce 2,3-phosphoglyceric acid. Both are same structure. So this 3-phosphoglyceric acid is the stable product. So it is known as C3 cycle. Clear? Hello?
Yes, sir.
Clear? So this is the first step. Then only we go for the full cycle. So this is the other intermediate step. Then how that ribulose bisphosphate produce 2-carboxy 3-dearabinol 1,5 bisphosphate, and how it becomes splitted into 2,3-phosphoglyceric acid. First it will produce a 6-carbon intermediate. First it produces 6-carbon intermediate. After that it's splitted into 2 different 3-phosphoglyceric acid. Up to this is clear now? Hello.
Yes, sir.
If anyone doubt now, please ask, because now I'll go in the cycle, directly in the cycle. Very carefully listen the cycle step. Then you will it is very easy to remember the C3 cycle. Now how? See, now by the steps I am showing like this way you try to remember. If you look at the book for the cycle, you will be very confused. So this while I am giving the presentation this is the very simplest way to remember. So all we know that Rubisco enzyme act on a substrate ribulose bisphosphate that is available within the stroma. Then what happened? The CO2 or bicarbonate is added and it produces 1,6-carbon intermediates 2-carboxy 3-keto-arabinotol 1,5 bisphosphate. That is the first intermediate, and soon after it will produce 2,3-phosphoglyceric acid, and the whole reaction is carried out by the enzyme Rubisco. And this first reaction is carried out by the enzyme Rubisco. So first step is substrate RUBP plus 3-keto plus CO2 produce 2-carboxy 3-keto-arabinotol 1,5 bisphosphate. This is a unstable intermediate, and these unstable intermediate soon after go hydrolysis and produce 2,3-phosphoglyceric acids. And this is the steps: ribulose bisphosphate first deprotonated and produce N-diolate intermediates. Then it produce beta keto intermediates, that means 3-keto-2-carboxy 1,5 bisphosphate. After that go hydrolysis and it again produce 3-phosphoglyceric acid in 2 double amount, because RUBP is a 5-carbon compound. Addition of 1 carbon, it becomes 6-carbon compound. After hydrolysis, 6 splitted into 2 3-carbon compound, that is 3-phosphoglyceric acid, which is a stable product, first stable product of this cycle. That's why it is known as C3 cycle. Clear? First step clear?
Yes, sir.
Next we go for the next step. What is the step? In this step, 3-phosphoglyceric acid will produce 1,3-bisphosphoglyceric acid. See, here 1 phosphate is there, here 2 phosphate. That means
Excuse me, sir.
Yes.
Hello.
Sharing the screen.
Sir,
So the PPT is
Hello.
not visible, sir. So only a
Not
screen
visible.
is visible.
Okay, I am again unsharing. Now it is visible.
no, sir.
No, sir.
Yes, sir. Visible.
Not visible.
Yes, sir.
Okay. First step is clear. This step is clear or not? Everyone, Dones, Sai Priti, Gayatri, Samiksha. This step is clear or not?
Yes, sir.
Again, I am saying that ribulose bisphosphate is available within the cell stroma, where CO2 first attached with that Rubisco enzymes. Process is known as carbamylation, and then it reacts with RUBP called carboxylation. After carboxylation, it produces a 6 carbon intermediate that is known as 2 keto 3 carboxy arabinotol 1,5 bisphosphate. This is the IPH number of the compound. This is a beta keto intermediate. After this is a unstable compound, and after product synthesis of this unstable compound, it immediately go hydrolysis and produce 2 molecule of 3-phosphoglyceric acid. So 5 plus 1 6, 6 split into 2 3 carbon. So this is the first step of that synthesis cycle. Clear?
Yes, sir.
Yes, sir.
Now go for the next step. What is that? 3-phosphoglyceric acid produce 1,3-bisphosphoglyceric acid by addition of the ATP. Who can say the name of what will be the name of the enzymes? If ATP is utilized, name of the enzyme is
Kinase.
very good. Now the enzyme name is the kinase enzyme. So what will happen? Another phosphate molecule will add here. ATP will split it down and ADP will produce, and phosphate will add it to that 3-phosphoglyceric acid. Now it become 1,3-bisphosphoglyceric acid. So it is exothermic and endothermic.
So it utilizes energy. So this enter of energy endogonic, sir.
Endothermic, very good. So next step is that utilization of, this is the reduction step. In this step, what will happen? NADH is used. If NADH is used, the name of the enzyme is dehydrogenase. You forget. Okay. If NADPH or NADH or FADH is used, the name of the enzyme is dehydrogenase. Okay. If NADPH or NADH or FADH used, so this enzyme will be NADP specific dehydrogenase. So in this, what will happen? That acid become aldehyde. The phosphoglyceric acid converted to phosphoglyceraldehyde, and this is the reduction stage. Next, the 3-phosphoglyceraldehyde immediately produced is isomer. What is that? Dihydroxyacetone phosphate. So now you remember that what is that? Phosphoglyceraldehyde, a 3-carbon compound, dihydroxyacetone phosphate, a 3-carbon compound. One is aldose sugar and there is the ketose sugar. Clear now?
Yes, sir.
That thing already you know. So phosphoglyceraldehyde and dihydroxyacetone phosphate. Which one is aldose and which one is ketose? Which one is aldose? Which one is ketose? Glyceraldehyde. Aldehyde is name is there, na? So aldehyde is the aldose sugar and acetone is the dihydroxyacetone is the ketose sugar. So this the enzyme will be the isomerase. Clear? So first step, second step, third step, fourth step. So remember, first RUBP to 3-phosphoglyceric acid, 3-phosphoglyceric acid to 1,3-bisphosphoglyceric acid, 1,3-bisphosphoglyceric acid to phosphoglyceraldehyde and phosphoglyceraldehyde dihydroxy acetone phosphate. Clear? This much clear?
Yes, sir.
Next step. Screen visible na?
Yes, sir. Visible, sir.
Next step. What will be the enzymes? When two molecules added and produce a one molecule, that is called aldolase. What will happen? That phosphoglyceraldehyde and dihydroxy acetone phosphate, those two isomer again add with each other and produce. See, this is also 3-carbon sugar and it is also 3-carbon sugar. So both 3-carbon sugar attached and produce a fructose 1,6-bisphosphate. The enzyme will be the aldolase. Next step is the fructose 1,6-bisphosphate one phosphate will removed and produce fructose 6-phosphate. What will be the enzyme?
Phosphorylation, sir. Phosphatase.
Very good. Phosphatase. So one phosphate removed means phosphatase. Addition of phosphate is the kinase. So this is the phosphatase. Now this fructose 6-phosphate will again produce fructose 6-phosphate. So glucose and fructose, all these are isomer. Glucose 6-phosphate and fructose 6-phosphate isomer. So that is isomerase. Again, glucose 6-phosphate to glucose, that is the phosphatase. So by this way, glucose will synthesize. Now, glucose plus fructose will produce sucrose and other starch, all other carbohydrate molecules they can synthesize by this way. Okay.
Sir?
So, hello?
So in the production of glucose from fructose, so the enzyme involved will be isomerase.
Huh. Obviously, isomerase. Because that two are isomer, na?
Yes, sir. Yes.
That do you ask the question? I told na, all other case we will see the different. Here we will see the different only. Glucose fructose.
Yes, sir.
So fructose 1,6-bisphosphate to fructose 6-phosphate. Now glucose is produced. See, if this reaction is going on, what will happen? The cellular RUB will be finished. If like this way reaction is going on, cellular RUBP will be finished. Now cell again has to regain the RUBP. Again, you have to regain the RUBP. For that reason, this next step is there. That is for regeneration of RUBP. See, this is the carboxylation, reduction, this is the synthesis, and now this step will be the regeneration of RUBP. So very easy. You remember first step is the fructose 6-phosphate to phosphoglyceraldehyde. This is 6 carbon. This is how much? Glyceraldehyde, how much carbon?
3 carbon.
3 carbon. 6 plus 3 equal to 9. It will split it into erythrose 4 carbon and 5 carbon. Erythrose 4-phosphate and xylulose 5-phosphate. By the enzyme transketolase. So what happened? Fructose plus phosphoglyceraldehyde, it will produce erythrose 4-phosphate and xylulose 5-phosphate. See, it is elose, xylulose, and is erythrose. One is the aldose sugar and there is the ketose sugar. Next step, this erythrose 4-phosphate. Again, see, remember up, down, up. First phosphoglyceraldehyde, then dihydroxy phosphate, then again phosphoglyceraldehyde. What will happen? Erythrose 4 carbon and dihydroxy 3 carbon. 4 plus 3 7 carbon. It will produce pseudoheptulose 1,7-bisphosphate aldolase. See, when two attached and produced, it is called aldolase. Pseudoheptulose 1,7-bisphosphate. Next, one phosphate will remove. Sorry, I this mistake. This will be the pseudoheptulose 7-phosphate. This will be pseudoheptulose 7-phosphate. Now, pseudoheptulose 1,7-bisphosphate, one phosphate will remove and will produce pseudoheptulose 7-phosphate. By the enzyme phosphatase, pseudoheptulose 1,7-bisphosphatase. Okay, this is the substrate. Enzyme is phosphatase. The full name of the enzyme will be pseudoheptulose 1,7-bisphosphatase. Now, see, up, down, then up. This reaction was with dihydrous acetate phosphate. Then it will again attach with phosphoglyceraldehyde and produce 7 plus 3 10. Now split into 5 plus 5. One is ribose 5-phosphate, and is the xylulose 5-phosphate. Next, this xylulose 5-phosphate produce ribulose 5-phosphate by the enzyme apimerase. By the enzyme apimerase. And ribose 5-phosphate also produce ribulose 5-phosphate by the enzyme isomerase. And then ribulose 5-phosphate will addition of 1 ATP, it will again produce ribulose bisphosphate or 1,5-bisphosphate. Now this is the again regeneration of RUBP. Clear na? Hello.
Yes, sir.
See, it is not so difficult, but see that book picture na, you will be more confused. In the book, the circle diagram is there, so it is very easy. Rubisco, RUBP plus CO2, 1,6 carbon intermediate, 2 keto 3 carboxy orbital 1,5-bisphosphate. It will produce 2 3-phosphoglycerate, 2 3-phosphoglycerate to 3-bisphosphoglycerate, then phosphoglyceraldehyde, dihydrous acetate phosphate, fructose or bisphosphate, fructose 6-phosphate. After that, it will produce glucose, and now first reaction phosphoglyceraldehyde, then dihydrous acetate phosphate, then phosphoglyceraldehyde. Now it will produce ribulose 5-phosphate, and again RUBP will regenerate. Clear? So this step is known as the carboxylation. Next step, this is the reduction, this is the regeneration, and this is the synthesis. So these are the different stages of CT cycle, and these are the enzymes. So I gave the name of the enzyme later because I know that you can easily can write the name of the enzymes. So now you understand now why I teach that enzyme structure, enzyme nomenclature, how to name, how type of reaction in the first. Hello.
Yes, sir.
Now it is clear now.
Yes, sir.
See, it is now it will be clear, but you have to again read this one. Today only you have to read, otherwise you will forget. 1 or 2 times you read the and try to draw the cycle at home. It will be very easy for you. This will be only 7-phosphate. Okay, so the first enzyme is the Rubisco. After the Rubisco, this is the 3-phosphoglycerate kinase. Then NADP glyceraldehyde 3-phosphodehydrogenase. Then this is the isomerase. Then aldolase, fructose 1,6-bisphosphatase. This will be isomerase. Then the transketolase, transaldolase, pseudoheptulose 1,7-bisphosphatase, transketolase, apimerase, isomerase, kinase. So this is the different steps of CT, all about the CT cycle. Clear? So again for recapitulation, for remembering, first step is the RUBP attached with CO2 and H2O produce 3-phosphoglyceric acid. 3-phosphoglyceric acid to 1,3-bisphosphoglyceric acid. 1,3-bisphosphoglyceric acid to phosphoglyceraldehyde or glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate and two isomer. One is the dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So what happened? That first glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This dihydroxyacetone phosphate and glyceraldehyde 3-phosphate attach with each other and produce fructose 1,6-bisphosphate. Then fructose 1,6-bisphosphate to fructose 6-phosphate. Fructose 6-phosphate attached with glyceraldehyde 3-phosphate and split into two compounds. One is xylulose 5-phosphate and erythrose 4-phosphate. Xylulose become ribulose 5-phosphate by the enzyme epimerase, and erythrose again attached with dihydroxyacetone phosphate and produce pseudoheptulose 1,7-bisphosphate. Pseudoheptulose 1,7-bisphosphate remove one phosphate group and produce pseudoheptulose 7-phosphate, and it again attached with glyceraldehyde 3-phosphate and produce two compounds xylulose 5-phosphate and ribose 5-phosphate. Xylulose again by epimerase, ribulose by isomerase produce ribulose 5-phosphate, which again converted to ribulose 1,5-bisphosphate. This is the summary of that C3 cycle. So these are the different steps of reaction I already told, and the name of the enzymes. This is the step-by-step reactions. Ribulose 1,5-bisphosphate to 3-phosphoglyceric acid, 3-phosphoglyceride to 1,3-bisphosphoglyceric acid, 1,3-bisphosphoglyceric acid to glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate to dihydroxyacetone phosphate by the enzyme isomerase, then aldolase, phosphatase, transketolase, again aldolase, again phosphatase, again transketolase, then epimerase, isomerase, and last one is the kinase. Only 13 steps of reaction. Okay, and these are the enzymes which which involved in different steps of that C3 cycle. First one is Rubisco, then 3-phosphofructokinase, then NADA, NADP, glyceraldehyde 3-phosphate dehydrogenase, triose phosphate isomerase, aldolase, and all they have the different function. Okay, so in summary, what is that? In 3CO2 will used to produce with 9 NADP and produce 1 glyceraldehyde 3-phosphate on carbon, and 2,6CO2 will produce 1 glucose molecules. Okay, clear? Hello?
Yes, sir.
Any doubts? Hello?
Yes, sir.
Any doubts? No doubt. Every day, no doubt.
So what is the process? So the name you told. So addition of carbon dioxide to enzyme is known as a carbo. Sir,
Oh, that one.
yes.
See, that things that you should not confuse. That is the maximum time people confuse. One is carboxylation and carbamylation. This one. This one. Carbamylation and carboxylation. You should not confuse. Carboxylation means, see, first step is the carbamylation. After that, when this CO2 will attach to that RUBP and produce what? 3-keto 2-oxy-arabinotol 1,5-bisphosphate. That step is the carboxylation step. Means this step. This is the carboxylation step. Means now that CO2 from the enzyme, by the enzyme attached with this and produce, this is step is carboxylation. So before carboxylation, one step is there, that is called carbamylation. Okay, this is takes place due to that some one active enzyme lysine present in the active site of the Rhodoscope. Lysine is a charged or basic amino acids in which CO2 will attach. This one.
Yes, sir.
See this name na you can write like that 2-carboxy 3-keto-arabinotol or 3-keto 2-carboxy-arabinotol. That is not the problem. You can write 3-keto first or 2-carboxy first, anyone, no problem. Because IUPAC name 2-carboxy 3-keto-arabinotol 1,5-bisphosphate. This is an unstable compound. This step you remember, okay na?
Yes, sir.
First step is Rubisco. Second step is that 3-phosphoglyceric acid, then 3-phosphoglyceraldehyde, then diet. See this way na you will very easy to remember. Otherwise, if you see the cycle na you will very confused where to start or where to finish. Okay na steps you can clear na steps. These are the steps with enzymes. See one another thing. Wherever the phosphate will remove na, we need water. I forget to give on water molecule. Here one water will be there. One water molecule will be attached there. Wherever phosphate will remove phosphatase, then you need to add one water. And whenever that ATP will be there, you must need Mg, magnesium. Okay, wherever ATP is there, you must need magnesium, and wherever that phosphate will remove, you must need water. So today all of you have to read the cycle, and if you don't you have any doubt now, tomorrow you will ask me. Okay, Lipsa. Lipsa,
Yes, sir.
are you getting or not?
Yes, sir. Getting.
Priyambada? Priyambada? Hello? Priyambada,
Yes, sir.
are you getting or not?
Yes, sir.
No. Don't say you saying in the yes and no, that itself denote that you are not understand. See, if you have any network issue now, you can listen later. You can download and you can listen later. From Impetus also, that video will come is coming on your site. You can again listen. Do you think I am going so fast the syllabus? Anyone? Hello? Hello? Prachi? Anyone has any issue that I'm going so fast so that you are not able to get that topic? Hmm? Is there any issue, other issue? You need the class, you're not getting, or you need any things? Hello? Telsini?
Yes, sir.
You need any other things? Anything that not getting or I'm going too fast?
No, sir. I'm fine with it, sir.
Rupeli, 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 Rupeli Pradhan. I think slept. Dones,
Yes, sir.
any problem?
No, sir.
Saurabh,
No, sir.
Gayatri, Subhasri. Any
No, sir.
problem now? Don't hesitate. Don't hesitate. You ask. Okay, then thank you. Tomorrow we study about the other cycle, that's C4 or CAM. Okay.
Thank you, sir.
Thank you.